And today we are here to hear oral presentations from students who achieve outstanding research work. Many of progress depends on research. The major revolution in life sciences over the past 2 centuries have reshaped the modern medicine. In this year, we have 47 students apply to this program through the university mostly. We selected through rigorous review by a committee of 80 professionals. After 3 months of lab work, 9 students to present the results today. To our students, I hope that this program inspires your passion for research and support your future career. Finally, today marks the 20 years anniversary. So I thank all of you and thank you very much.
Good morning, everyone. Welcome to the conference of summer research program for medical students. I'm very pleased to chair the first section. My name is P.C. Yang from National Taiwan University. First speaker is Chao Yu Zheng. He's post-year grade medical student from Chang'an University. Advisor, Professor Liu Shen. Please.
Transient SASP can be beneficial. We apply a strategic screening by intersecting tumor genetics-associated genes with 4 clinical outcomes. The spatial map showed that DRG5 is predominantly expressed in the tumor region and analyzed the global gene expression changes to see which pathway were affected. BDNF is a well-known secretory protein, forming an autocrine group that enhances the cell anti-apoptotic ability. It is well established in previous study that NF-kappa B acts as a master regulator of the SASP.
And my question is, because DLG5 is a polarity protein, I'm wondering when regulating the downstream target senescence secreted protein, the location of DLG5.
Based on our seniors' previous experiment, they found that the DRG5, when it is triggered by the interferons, it will actually goes to the nucleus type, it will enters the nucleotypes.
TNBC is defined by its absence of estrogen receptor, progesterone receptor, and HER2 receptor. So this figure represents the epidermal growth factor, which is EGF signaling pathway. For cancer cell initiation, it will inhibit cancer cells by degrading the damaged organelle and protein. Summarize that TNBC cell has a higher EGFR expression and more active basal autophagy level. So we may conclude that EGF-induced autophagy will promote migration in TNBC cells. And the p62 is decreased in the control group, and it is accumulated in the LG5 knockout group.
I think maybe you want to extend your study. So have you checked whether alternation of autophagy also affects cell invasiveness?
So if we use the LG5 knockout group, can only study how autophagy affects its migration, but it is crucial for me to understand more how it affects the mechanism for its migration. Yeah, it's our future work.
In addition, aberrant glycosylation also contributes to RA pathogenesis. Moreover, in RA patients, IgG glycosylation is also altered. In CIA mouse model, P19 deficient mice fail to develop disease. Loss of IL-23 increases sialyl transferase expression in B cells. We found that in MGAT5 mutant mice, the disease onset is accelerated at about 3 to 5 weeks of age, downregulating MGAT5 dependent unglycan branching on CD4 T cells.
Okay, so have you just looked on the antigen specific for that, or you think it's a global CD4 effect, not specific to the antigen of the B cells?
Use flow cytometry to do LPH staining to see whether this increases the unglycan branching on CD4 T cells or antigen-specific T cells.
That means that we also increase some other autoimmune phenomenon.
In B cells, the MGAT5 expression is lower in P19 knockdown mice, and further investigation also shows that in MGAT5 mutant mice, B cell contribution might be minimal compared to T cells. In contrast, in non-allergic asthma, the number of ROR gamma T plus IL-3 is increased. And this tells us, this may explain why obesity has long been considered to worsen inflammation of airway of type 2. What will happen if our lung is in high lipid level, such as we are obesity or in high-fat diet? The cytokine which is secreted by IL-C2 like IL-5 and IL-13 will depress. This finding tells us that we can use PLN2 to change how IL-C2 reacts.
How about the outcome? Because I didn't see any asthma activity in the mice with normal diet and with the high-fat diet. Did that really change the outcome of asthma?
Produce more GATA3 plus IL-C2, and it will have a lot of cytokines such as IL-5 and IL-13. In our data, IL-5 and IL-13 will decrease.
We would like to welcome Professor Zhou Yuting to be our moderator.
This is Yuting Chao, actually from Tsinghua University. Okay, our next speaker is Sun Pei-En. Pei-En is from National Yangming University, and his professor is Professor Yang Zhiyu. Welcome.
Around plaques and tangles, microglia, astrocytes, and infiltrating T cells interact with the neurons and drive neurodegeneration. Most research has been focused on MSC exosomes, together with pluripotency assays at the top. To link these functional effects to exosomal cargo, we perform small RNA sequencing on iPSC exosomes and UCMSC ones. Utilizing AD-focused annotations from GO, KGG, Reactome, and MIEAA, which we plan to follow up in functional assays.
Okay, so your exosome treatment reduce the phosphorylation, right? If you want to explore the link or the mechanism of the effect of exosome, how would you design your result, your approach?
We use orthogonal treatment with differentiated neurons, and then we add iPSC exosome to see what if the effect is what is the effect that induce the survival of the neurons.
This is the next 3 slides will show the flow chart of our study. So after the inclusion and exclusion criteria, we have patients who with type 2 diabetes who newly initiated the study drug classes between the study periods, and around 500,000 patients in GLP-1 receptor agonist versus DPP-4 inhibitor group. And here is the sensitivity analysis of our study. with SGLT2 inhibitor and GLP-1 receptor agonist versus DPP4 inhibitor persist, which further support our main finding.
Can you speculate why those SGLT inhibitors or GLP-1 receptor agonists that provide us reduction of COPD incidence?
From some inflammatory process in our body and also some vascular process. So we find that some study that has focused on discover the diabetes drugs on the effect of inflammatory process, and some biological study have found that they reduce the inflammatory process of our body.
And from day 0 to day 10, we repeatedly changed their ears using the 0.2% oxazolone or the vehicle control in the groups. And compared to the normal control groups, the oxazolone treated group showed severe erythema, swelling, and scaling. And also under toin blue staining, we checked the mast cells in the dermis. So this suggests that the Langerhans cells might migrate into the draining lymph nodes from the epidermis.
A good presentation. One short question. You proposed that internalization is one of the possibility in your paradox finding, right?
Yes.
How to prove it?
There's a report in Immunological Journal that the internalization of c-KIT shown in their presentation, and they did experiment to try to upregulate the SCF in their dermal tissues and check that the mast cells with c-KIT will be downregulated.
Other pathways such as glutamine signaling and HBA axis also implicated in depression. Carriers with CCR6 variants may experience a higher depression improvement. CCR6 variants may alter how Th17 cells respond to 5-FU. As a receptor involved in brain signaling, GRM8 may contribute to the communication along gut-brain axis. We found that TS-based chemotherapy was associated with greater improvement in depression symptoms.
Do you include the one very important clinical variable for cancer patients is the response to treatment, and as a confounding factor for this study, because chemotherapy response may influence patients' emotional status severely.
I did not put this factor in the analysis. This is an retrospective observational study and missing data in our database. So to simplify this research, we first built this workflow for analysis.
we start from the Excellent Award. Mr. Yu Rongchen from National Taiwan University. Outstanding awardee is Zheng Chaoyu. The top grade awardee He Yu Huang.